Hello! Again, I didn't manage to appear on the screen to say a few words. Couldn't organize it. I will do that later in this series of videos. But now, when we say genes are the basic units of evolution, or genes make us, what are we really talking about? What is a gene? A gene by any other name. Pinning down exactly what a gene is turns out to be no easy task. A geneticist may define it as, in inverted commas, the basic unit of heredity. A molecular biologist as, in inverted commas, a peptide encoding length of DNA. Dawkins defines it as, in inverted commas, the fundamental unit of natural selection, i.e., the replicator responsible for evolution on Earth. A quick review of the biology involved may clarify the issue. All, or almost all, life on Earth is built on DNA. A footnote. I say almost all for two reasons. Firstly, some viruses use another molecule, RNA, to encode their genetic information. There was a long debate in biology about whether viruses should be counted as living things. All that this argument really showed was that there is no black and white definition of, in inverted commas, living. How complicated must a reproducing molecule be before it is considered alive? But I digress. The second reason is that arrogance should not let us preclude the possibility of life evolving on other planets. Dawkins and I both agree, I think, that although such life would come about by a process of evolution through natural selection, there is no reason to suppose that the replicators or genes involved should be composed of DNA. Close the footnote. DNA is a long but simple molecule consisting of regularly repeating subunits which encode the genetic information. Just as a computer disk is essentially a long series of data split into different files, so a single DNA molecule may have many functional genes encoded along its length. Unlike the binary system of computers, however, in which every, in inverted commas, bit of data is represented by a 0 or a 1, DNA uses 4 different chemical compounds called nucleotides. These are usually written A, T, C, and G using the first letters of their chemical names. If you, in inverted commas, read the sequence on a computer disk, you may get 100010011110, whereas a DNA sequence would look like ATTCG, ATTCG. When DNA is sequenced, the result is a gel with 4 letters marked in correspondence to the 4 letters of DNA's alphabet. Distance along the DNA molecule is represented vertically, and horizontal lines in each column show which letter is at that position. I mention this only because it is one of the media's stock images when discussing the matter. In general, the information encoded by DNA is used for making one thing: proteins. A footnote. Again, my proviso is to cover my back. Much of the DNA in higher organisms does not encode proteins. This apparently junk DNA has recently become the focus of much study, as many believe that it serves important structural and regulatory roles for the genetic machinery. If this is true, then this DNA has, of course, been designed by natural selection, just like its, in inverted commas, more functional cousins. However, as it does not encode proteins, its effects will be limited to its immediate cellular environment. Close the footnote. These proteins not only make up the organic structures of which you and I are composed, they also regulate in precise detail the workings of the cell, and hence the organism. They control chemical reactions responsible for everything from digesting your food to repairing damaged skin. It is through proteins that DNA makes its mark on the world. Change the DNA sequence and you change the protein sequence, thus the effect it has on the organism. In inverted commas start and in inverted commas stop signals ensure that the cell, in inverted commas, knows where protein encoding regions begin and end. Molecular biologists call this functional unit a, in inverted commas, cistron, to avoid the problems of the ill-defined gene. To distinguish between the information carried on DNA and its effects, biologists use the terms, in inverted commas, genotype and, in inverted commas, phenotype. Suppose you carry a gene for blue eyes. Your genotype describes the DNA involved, while the phenotype, in brackets the gene's effects, is blue eyes. Footnote. As with most characteristics, eye pigment is in fact controlled by several genes, as several proteins are required to produce the necessary compounds. So how can I validly talk about a, in inverted commas, gene for blue eyes? The essential point is that it is a gene for blue eyes, in italics, all things being equal, an issue examined in greater detail later. Close the footnote. The complete genotype of an organism, i.e. all its genes, is called its, in inverted commas, genome. In many organisms, the genome is split between several long DNA molecules, in inverted commas, chromosomes. In our computer analogy, this corresponds to having several disks of data. Humans have 23 pairs of chromosomes, in which each pair functions as duplicates. Suppose our eye pigment gene is located at a certain position on chromosome 3. This means that there will be a gene for eye pigment at the same position on both copies of chromosome 3. However, the exact DNA sequence may differ between the copies. One may encode a blue pigment, the other a green pigment. These variations are called, in inverted commas, alleles. So the gene for eye pigment may possess a, in inverted commas, blue allele, a, in inverted commas, green allele, and so on. Usually, alleles differ from each other by just a few nucleotides. Whether the individual ends up with blue eyes, green eyes, or something in between depends on the gene and alleles involved. In actual human eye color, the brown allele is, in inverted commas, dominant to blue. So an individual with one copy of the blue gene and one copy of the brown will have brown eyes. Blue is then said to be, in inverted commas, recessive to brown. An individual with two blue alleles would, of course, have blue eyes. The relationship between other pigment combinations is somewhat more complicated. In computer terminology, this system is equivalent to having two of each disk, where the same files are present on each copy in the same place. The exact content of the files, however, may differ by small amounts. Eggs and sperm, in brackets sex cells, contain just one of each pair of chromosomes, thus just one of the two possible alleles. These combine with your partner's sex cells after fertilization to produce the full complement of 23 pairs and hence a new genome, your offsprings. The only slight oddity is the sex chromosomes called X and Y. Although a woman's two X chromosomes form a matching pair, the XY combination in men doesn't. The Y chromosome is significantly smaller than the X chromosome and thus lacks a large portion of its genes. They separate in the usual way when sex cells are formed, which means that sex is determined by the male's genes. An egg must contain an X chromosome, whereas a sperm can contain either an X or an Y. The last complication in the process is that before the chromosomes are bundled off into sex cells, DNA can be, in inverted commas, swapped between the pairs in a process called, in inverted commas, crossing over. So instead of giving your offspring one or other chromosome, you may pass on a chromosome composed 1/3 of one copy and 2/3 of the other, for example. In our disk analogy, this is equivalent to swapping some of the files on one disk with the corresponding files on the other. In this way, two new disks have been created, composed of parts of the former pair. In most species, which parts of the chromosomes cross over is fairly random, so a new unique chromosome tends to be produced each time. Of course, if the chromosomes in the original pair were identical, in brackets, i.e. they had the same alleles for each gene on the chromosome, close the bracket, crossing over would produce no net change. In practice, however, there will be differences in many gene locations. Thus, each sex cell will inherit a unique set of 23 chromosomes. Armed with this knowledge of molecular genetics, we can now make sense of the definitions of a gene. To most biologists, the gene is a functional piece of DNA which encodes one protein, in brackets, roughly the sense in which I have been using it until now, close the brackets, a cistron. Dawkins, however, adapts George Williams' definition and sees a gene as a quotation, any portion of chromosomal material that potentially lasts for enough generations to serve as a unit of natural selection, close the quotation. We have seen that replicators must persist, in brackets, albeit as copies, for many generations, if they are to act as a target for natural selection. Thus, chromosomes themselves cannot be replicators. They are chopped up and reformed in each generation, thanks to crossing over. Parts of chromosomes may not necessarily be destroyed in this way. They may survive many generations before being split. As the process is more or less random, the smaller the length of chromosome, the less likely it will be destroyed, and the more generations we can expect it to survive intact. This is the first problem, for crossing over does not respect the boundaries of cistrons. Bits of files can be swapped with corresponding bits on the other disk. Thus, eventually, we should expect any length of, in inverted commas, chromosomal material, no matter how small it is, to be split in two, never again to meet. Even a single pair of nucleotides will eventually part company. This point is made by Mark Ridley, a quotation. If we take a long enough view, the only finally permanent units in the genome are nucleotide bases. However, this long view holds little interest for us as we are concerned with the time scale of natural selection. It takes 1,000 or so generations for a mutation's frequency to be significantly altered, and over this time, genes, but not genomes or phenotypes, will be practically unaltered. Close the quotation. This is why Dawkins uses the phrase, in inverted commas, enough generations in his definition. The gene is that part of a chromosome which persists unaltered long enough for selection to influence its frequency. So, of course, the gene is the unit of selection. It is defined to be exactly that. A gene is thus a vague entity. We cannot easily identify a piece of chromosome as being a gene in Dawkins' sense unless it has proved its persistence over time. Some, in inverted commas, genes may in fact consist of many sequential cistrons. Later we will examine whether or not selection acts just at this genetic level. Are individuals, groups or species possible targets for evolution? Are they true replicators? If we find that they are, perhaps we would be tempted to call them genes. Dawkins is clear, however, that while he does not pretend to have a precise physical definition of the gene, it is a length of DNA lying somewhere between the cistron and the chromosome. Ridley again summarizes the point. Quotation. Williams defined the gene as, in inverted commas, that which segregates and recombines with appreciable frequency, close the inverted commas. According to this definition, the gene need not be the same as a cistron. Rather, it is the length of chromosome that has sufficient permanency for natural selection to adjust its frequency. In practice, the replicator, in brackets, or Williams gene, close the brackets, does not consistently correspond to any particular length of DNA. Close the quotation. Thanks for listening. Bye.